Welcome to the Future Tech edition of the Finding Genius podcast. Forget frequently asked questions, forget common sense, common knowledge, or Googling for information. How about advice from a genius in their field instead? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond. They become very good at what they do, but only 0.1% are the geniuses of their profession. Richard has made it his life's mission to interview the geniuses of their fields in areas such as AI, 3D printing, quantum computing, blockchain and Bitcoin, and more. Don't miss out on amazing podcasts with geniuses. Review us on iTunes or wherever you listen, and go to futuretech.findinggeniuspodcast.com and subscribe today.
Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Vittorio Sebastiano. He's the co-founder and scientific advisory board chairman of Turn Bio. Website is turn.bio. We're going to be talking about potentially reversing aging by reprogramming cells. So, Vittorio, thanks for coming. How are you doing?
Yeah, hi, Jacobs. Pretty good. How are you?
Pretty good, yeah. Well, my cells could use some rejuvenating, but let's see what's in store there. So, tell me about Turn Bio and about your work there. What's the premise of Turn Bio?
Sure. What we do at Turn Bio, so the principle, the idea behind the work that we do at Turn is that it is possible to reverse the epigenetic landscape of the cells and bring it back in time so that, a cell which by the process of aging becomes dysfunctional with time can actually be reprogrammed or reversed in a way that now it becomes more youthful, more functional, and this could potentially have repercussions on the cell itself, but also broadly speaking, also systemically in the individual. So the basic principle that we apply is that we believe that fundamentally aging, the cellular level, happens because there is the so-called epigenetic drift. So this epigenome, which is, broadly speaking, refers to a program in the cells that regulates or controls what genes a cell should express or should not express, becomes dysfunctional with time. And this is, it's not necessarily the primary cause of aging. It is possibly, a secondary cause of aging, which is triggered by other factors, pollution, inflammation, damage of any kind. But these damages, result at the end of the day into a change in the way this epigenome is organized and regulated. And in the long time, this progressive accumulation of mistakes lead to a dysfunctional cell type, which explains aging. So what?
All right, Vittorio. Yeah, from what I understand, epigenetic changes are an adaptation of an organism to different changes. So if I live at, let's say, a high altitude, I would probably experience some of my genes upregulating and downregulating to accommodate. Or if I live in a hot climate, or if I have a certain diet, so it's an adaptation of the organism as it lives, and that's part of epigenetic change, right?
Absolutely. So the epigenetic programming is actually kind of I think it was a very, it doesn't exist in all the organisms. Methylation, for example, doesn't exist in bacteria, for example, and it's actually an evolutionary trick that actually I think allowed multiple species actually to react in real time to changes in the environments, because it's really like this analog type of program that is very malleable, very elastic, very plastic, and can rapidly respond actually to the changes in the environment. Now, for the most part,
what does epigenetic change look like on a molecular level? I've heard about for instance, methylation. I've heard about changing of histones.
Yeah.
In your view, what are some of the ways that epigenetic change happens, and what does that do? Just give me a basic overview.
Sure. Yeah, well, there is different mechanisms of epigenetic regulation. So one of the most obvious, the known, is the methylation of the DNA. Methylation can occur at the level of the cytosines, which are bases in the DNA, and these changes of the of these bases, can modulate, for example, gene expression. So they can turn off or turn on specific genes in response of the how many of those cytosines are methylated or unmethylated. But that's only one example. There's actually way more. For example, the histone modification. So histones are proteins around which the DNA is wrapped. And so the positioning of the histones on the DNA, on the sequence, but also specific modifications of those histones can regulate gene expression. Again, they can turn on or off genes depending on the on how these histones are positioned and or modified. But actually, there is way more than that. For example, even the organization of the chromatin, so the organization of the DNA within the nucleus, is an epigenetic trait. So broadly speaking, epigenome is basically a layer of regulation which does not depend on the sequence. That's what epi means. It means beyond. It means beyond the genetic. So it's a regulation mechanism that is not dependent on the sequence of the DNA, but it's dependent on other modifications which change and modify the DNA and the chromatin in time and space.
All right. So from my understanding, epigenetic changes will either upregulate or downregulate genes, meaning that the gene will express more or express less, sometimes not at all.
Yeah.
What about the underlying DNA? Have you observed, or has it been observed, that epigenetic changes can reshuffle the underlying DNA, cut out pieces or add pieces to it? Or is it just the underlying DNA is kept and it's just how the genes are expressed has changed?
Fundamentally, it's how the genes are expressed. Now, this though can happen in different ways. So as I said, one way, a very simple way of looking at it is a gene is on or off. Then there is like how much. So when it's on, how much is it on? And so there is also kind of nuances of epigenetic regulation that can regulate how much a gene is activated. So it can be very highly activated, or it can be just expressed at very low levels. And again, epigenetic changes can regulate are actually the main regulators of this behavior. But then there are also a gene can be on, off, or highly activated, or lowly activated, depending on where it is in the nucleus. And again, the epigenetic changes and the 3D chromatin organization can regulate the positioning or the structure, the structural features of the gene, or of the niche where the gene is located, and this can actually influence how much a gene is activated or inactivated.
Okay, so what happens when throughout someone's life, I figure they acquire epigenetic marks? How do you tie this into the appearance of disease or senescence of cells for getting old? What patterns have you seen in people that, as they age, maybe their marks are piling up in a certain way? Like what have you seen?
Yeah. So in complete honesty, actually, we are just starting to understand what aging means at the molecular level and at the cellular level. I mean, we obviously know that aging occurs. We see it happening, every day when you look into the mirror. But really, at the molecular level, at the cellular level, what this means, we haven't completely understood yet.
Well, what do we understand? Are we seeing certain patterns? Is there okay, so what are you seeing, and how is that correlated?
So we do see patterns. For example, as I said before, we see that with time, the set of genes that has specific cell type. So let's just make an example. Neurons, for example, in the brain. We know that with time, the neurons, and this applies to any other cell type in the body, we know that with time, the cells lose the ability to properly and correctly express the set of genes which are supposed to be expressed in that cell type. So the identity of the cell remains the same. So a neuron, in a 10 years old individual, or a neuron in a 90 years old individual is still a neuron. Okay, so there is a fundamental epigenetic profile that has not changed so much to explain, a change in cell identity. So again, the neuron is still a neuron. The problem is that the neuron in an older individual is dysfunctional, meaning that it expresses genes that are not supposed to be there, or it does not express genes that should be there. And what we are understanding, we are learning that actually this process can be triggered by different phenomena. As I said before, inflammation, for example, increase in reactive oxygen species, damage of any kind. But fundamentally, this results in a change of the epigenetic profile of that cell. So with time, some small epigenetic changes occur, and they kind of accumulate over and over and over. But at some point, you reach a threshold where they are no longer sustainable, and so the cell now becomes dysfunctional. And it can become dysfunctional in many different ways, of course. One of the extreme is that it may turn, for example, into a carcinogenic cell, or it can become a senescent cell, or in most of the cases, it doesn't become senescent, it doesn't become carcinogenic, so it is still the same cell, but it's just dysfunctional. It doesn't express the proper set of genes that should be expressed there. So what we believe is that by understanding this mechanism better, we can actually leverage on that and reprogram those changes and bring them back in time to a more functional and youthful state.
One looked at I know that in cancer, cells will have different genes, or not different, that are mutated. But what about the epigenetics of cancer cells? Has anyone looked at that and done a comparison to non-cancerous cells?
Yeah, people are, absolutely. They're looking at that, and it is interesting to also realize that when you look at the mutations, of course, the cancer is a much more complex in a way disease, but it's very interesting for example, to realize that some of the early mutations, and this seems to be true across different cancer types. Some of the first the first mutations that occur and that can eventually lead to the formation of a tumor occur actually in genes that are responsible for the epigenetic regulation of the cells. So, for example, DNA methyltransferases. So these are enzymes that regulate the methylation of the DNA, or the TET enzymes. Again, these are enzymes that are important to regulate nuances of the methylation profile of the DNA. Histone modifiers, for example. So these probably are the first mutations. So once the epigenetic regulation becomes dysfunctional, and in the context, of course, of cancer, is triggered by a mutation, by a genetic mutation, then you have the progression of the cancer and you have the progression of the disease. With aging, it's not necessarily a mutation in the gene, but it's a progressive accumulation of random mistakes in the epigene.
Provide a clue then if some of the genes involved with epigenetic changes are the first to mutate or the first to go. Have people observed that cancer, for some reason, it's more epigenetically fluid? Does it tend to acquire marks or remove marks quicker than normal cells, or in a different way?
Yeah, absolutely. That's actually known. Again, the granularity of these hasn't yet been fully understood, but it is absolutely obvious that from an epigenetic standpoint, and also then from a metabolic standpoint, the cancer cells become in a way more youthful, and that also explains why they are so aggressive, they proliferate so efficiently, and they kind of occupy niches in a very, very efficient way. So yes, I think there is also a lot to learn by understanding that process and, kind of leveraging that knowledge and repurposing it for a different question, which is, a more physiological aging or rejuvenation, if you will.
How do you determine that a cell is more youthful? What tells you that, from an epigenetic standpoint? Does it just have fewer marks, or is it the pattern of the marking, or what tells you that?
Yeah, so that's thank you. That's $1 million question, in the sense that, well, when we started this work about 5 years ago, I was stunned by and very surprised by the fact that there hasn't been yet kind of a unifying theme to explain aging, and in retrospect, probably that makes a lot of sense. So with the exception probably of the methylation clock that we can probably touch upon later, there, and by the way, even in that case, there is not yet kind of a deterministic, in a way, picture of that phenomenon. What we see with aging is that there is a number of changes that occur with time. So there was a nice review that was written by Manuel Serrano and others that defined for the first time the hallmarks of aging. And I think that was a very, very great work, because it really helped us kind of define in a very broad way still, but define, this process of aging. And basically what they proposed was a number of hallmarks, from senescence to epigenetic changes, nutrient sensing regulation, stem cell homeostasis, and many more, mitochondrial dysfunction, hallmarks that altogether explain age. Now, some cells may age, for example, primarily with regards to some of these hallmarks. So it's not necessarily true that the cells, as they age, change their profiles of across all the hallmarks. So that this is a very important thing to keep in mind. But they do. To some extent, they do across some or sometimes all of these hallmarks. So when we define aging at the cellular level, I think we have to be very, very careful and be agnostic of the fact that we should probably be looking at all these hallmarks, and some may change, some may not. And when we define the rejuvenation, we have also to holistically and comprehensively look at all of these hallmarks altogether. Methylation changes, are definitely one of these, epigenetic changes are definitely one of these hallmarks. And some people, have developed, for example, methylation clocks that can predict the chronological or biological age of a sample or of a cell type just by looking at the methylation profile of a cell. Now, we don't understand yet if those changes are deterministic. So we don't know if those changes are causing aging or if they're just correlated with aging. So we don't know that yet. And we don't know if there are specific hotspots in the genome where, if a methylation, change occurs, then this triggers a number of processes, down the road. We don't know that yet. We just know that, those changes occur. And when we see them happen, then we can define that cell, aged or older in a way.
So what are some of the correlations you see in senescent cells? I mean, well, here's a question. So is senescence broken up into different stages, or I don't know, is it like a monolithic thing? Or again, is it broken up somehow and characterized into different stages, or I don't know, different aspects or phenotypes?
Yeah, well, the first distinction that I would like to do, Richard, is the fact that senescence is one aspect of aging. It's not necessarily aging per se. So as our body ages, some of the cells enter senescence, but it's only a small fraction of the cells that enter senescence. And when I say senescence, I mean replicative senescence. So, the cells at some point accumulate a number of epigenetic, but also genetic damage. For example, telomere attrition, telomere erosion. And this basically leads to the fact that the cells stop dividing. And this, in a way, is actually good because, this mechanism prevents, for example, the cells, or it's kind of a QC, a checkpoint the cells are so damaged that it could actually go down the cancerous kind of path. Senescence actually is a mechanism to prevent that. And it has also been shown that senescence is probably a mechanism or a phenomenon that helps the regeneration of tissues in very specific context. Now, the downside, though, of senescence is that, these cells, of course, they stop dividing, and again, for good reason, but then they start secreting, the inflammatory cytokines and so forth. But again, I would like to remind everybody that this is just 1% of the cells in an older body. The vast majority of the cells in an older body or in an aged body are aged, but not necessarily senescent. So this is a very important distinction that I would like to make, because our technology, for example, could complement existing technologies that are already out there. For example, senolytics. So with senolytics, basically the idea is that you provide a molecule, a drug, or whatever. You specifically target the senescent cells. So again, these 1 to 5% of the cells in the body. You clear them out, and so by doing so, you would reduce the inflammatory cytokines, and this could have a systemic beneficial effect on the body. But that doesn't make it makes the body more functional in a way, less inflamed. It reduces inflammation, but it doesn't make it younger by all means.
Of all the cell types in the body, in the human body at least, which cells are easiest to work with to understand senescence? Do you pick ones that are long-lived and therefore maybe slowly come to senescence, or you look at fast-dividing ones that maybe have a very short life cycle and show, or are there cells that, for some reason, in people or mice that show senescence first, early on?
No, I would say that senescence senescence happens in all the tissues at different degrees. Some tissues are less prone just because of the way they have been designed over time. For example, the intestine, it's highly proliferative, and so there is a reduced degree of senescence in the tissues where there is less proliferation, the cells, there is a higher percentage of senescence. And then there could be situations, like, for example, fibrosis or, heart failure. There could be some context where, senescence, just because of the damage that has been induced, is a very predominant phenomenon, specifically in that tissue. Now, these studies in vivo, of course, have many have been done, but it's really difficult to understand. So senescence primarily has been studied in vitro, and so there is ways to induce senescence by DNA damage or other means, and so this is kind of a proxy in vitro of what happens in vivo. And so all we know primarily about senescent cells comes from in vitro studies where we induce this phenomenon. And senescence also naturally occurs. So if you take cells from an individual and you just grow them up, over time they reach these the Hayflick limit, and that this obviously what was described by Hayflick, and at some point the cells, they exhaust just because again their telomeres shorten, and at some points they don't cycle anymore just because they can't. At that point, they enter replicative senescence. So that's kind of a more natural form of senescence, if you wish. But again, it's an in vitro model of senescence. Right now, people are looking into, in vivo models of this. And so all the senolytics, for example, work has been done with some animal models of this.
So in the world of senescence, how big of a role are epigenetic marks, do you believe? A tremendous role? And if so, is that really the focus of Turn Bio then, is to establish what correlations there are between epigenetic marks and senescence? Or how would you put it best? No,
actually, what I would say is that we are complementing the work of anti-senescence approaches. Actually, the senescent cells are so much damaged that you don't want to reprogram them. You don't want to bring them back into the game. Actually, the way we are thinking about this problem is that, again, maybe with senolytics, we can clear up the senescent cells, so get them out of the equation, and then reprogram and rejuvenate the cells that are still there, that are non-senescent, so they're still replicative, but they're just dysfunctional. Again, reprogramming the senescent cells would be probably a very bad idea, and that's definitely not what we want to do. We want to get them out of the equation and then rejuvenate and epigenetically reprogram the cells that are still functional, that are still there, but they're just dysfunctional from an epigenetic standpoint.
Well, at least we thought of one good thing and what not to do. So in terms of the reprogramming, what does that look like? What does that mean?
Yeah, so the reprogramming, again, this is a very broad term that is used with different meanings in different contexts. So just to clarify a little bit. So broadly speaking, reprogramming means changing one cell type into another cell type. So this is how typically is used in the scientific community. So for example, reprogramming a skin cell into an embryonic-like cell, or reprogramming a skin cell into a neuron, or into an hepatocyte, and so forth. So primarily, people think about reprogramming as a change in cell identity. The way we think about this, it's not a change in cell identity, but it's a change in cellular age. So as I said before, the same way the epigenetics explain why a cell should express a set of genes and not another set of genes, the same way, in a very similar way, basically, epigenetic explains why the same cell type changes expression profiles over time. So our reprogramming is a reprogramming of the epigenetic signature of age. So the core principle here is that by controlling, tightly the time and the duration of the reprogramming, we can erase those epigenetic mistakes that have been accumulated over time, and we can make a younger version of the same cell type without changing its identity. This is very important because it's actually, one of the fears that some people have, that, by pushing too much this reprogramming, you could actually change the identity of the cells. Well, our data demonstrates so far that if you know the biology of that specific cell type or tissue, and if you know exactly, the time that's required for the cells to rejuvenate but not change cell identity, so that's really the key, and that's exactly where that's where our technology, is powerful, because we understand that process and we don't push the boundaries too much.
What does the process look like? I know you can't say some of it's proprietary. That's okay.
Yeah, well,
within the realm of what you can say, what is the process like?
Oh no, it's something that I can definitely share with you. So we obviously the vision, the big vision, is that we will be able one day to do this in vivo, without extracting the cells from the body. Again, this is our long-term goals, and we already have a plan actually to tackle this goal. But in the short term, the way we're doing it, or the way we're envisioning it, is to do this therapy ex vivo. So we isolate the cells, whatever cell type. What we have a couple of indications that have already been prioritized. But the idea is that we isolate the cells from the body, from the individual, the patients. We rejuvenate them in vitro, and then these cells go back into their original tissue or niche. We have shown this to be extremely powerful, for example, for muscle stem cells. So when we extract the stem cells, the muscle stem cells, from the skeletal muscles, and we treat them for just 2 days, 48 hours. So it's very short. And we put them back into animal models, we see that we can basically restore the muscle function of a young mouse. We're talking about 30 to 40% of a force induction, which is pretty remarkable. If we can do this with stem cells, now since stem cells have these, are naturally endowed with this ability to self-renew and to differentiate into the cells of the tissue, if we can do this with stem cells, now we have a very long-term effect. And we have shown this in mice. We have seen an effect of the rejuvenation all the way up to 2 months after the treatment. And again, 2 months might not sound too exciting, but 2 months in a lifespan of a mouse is about, it's a lot. And so it's probably 15 to 20 years, when it comes to human lifespan. So it's pretty significant. Of course, we need to do more work. We need to show that this is really applicable and safe. There's obviously a ton of work to do, but preclinical data and preclinical studies show that actually this is pretty remarkable and efficient.
Literally, what have you done to the muscle cells to make them more youthful? And again, how do you know, okay, they're more youthful now? Like, what tells you that? What kind of, what's changed in the cells?
Yeah. So the treatment is very simple. So we extract the stem cells from the muscle. And the good thing is that we actually have we have two patents which are exclusive to Turn. So one is obviously the one on the epigenetic reprogramming of aging. And the other one is a patent that was developed by one of the three co-founders of the company, Marco Quarta. He was working as a postdoc back then in the Thomas Randall lab. He's an expert in aging and muscle biology. And so, long story short, Marco and Tom developed a way to extract the stem cells from the muscle and keep the stem cells quiescent, so preserving, in other words, preserving their stemness ability. By combining that technology, again, which is exclusively licensed by Turn, with the epigenetic reprogramming of aging, which is just a very short reprogramming of 48 hours, by combining the two and by transplanting then the cells that, presumably rejuvenated back into the mice, we have shown that actually there is an increase in muscle regeneration. So the cells now are behaving like youthful cells. So they regenerate the muscles much faster. They form more fibers and thicker fibers. And so the entire volume of the muscle is rejuvenated. From a physiological standpoint, they're also juvenile. So they have a higher mitochondrial functionality, more ATP production. They cycle better. But most importantly, they do not form tumors. So the only thing that they do is just they form more fiber. They form muscle fibers, more and thicker, but they don't form any other form of, dysplasia or cysts or tumors. And this is very important because, again, it speaks for the safety of the approach.
Do they continue on their path of normal aging once you've set them back, restored some of their youthfulness? Do they just normally age from there, or do they seem to age differently?
Well, we have not done a comprehensive set of studies in that regard. What we know is that 2 months after, as I said, if we go back to the same mice and we induce a second injury in the muscle, we see that actually the cells, regenerate the tissues in a very, very similar fashion as if they were very young. So there is a memory of that process. Now, quite honestly, my hunch is that yes, they will age because of course they are in an age organism, and aging is a very physiological process that occurs. So they will age. We don't know yet if they age at a different pace or if somehow they are a little bit more resistant to that process, or they're just brought back in time, and then their clock for them starts at an earlier starting point. So we don't know that yet. So of course we need to do more studies, but we do see that at least 2 months after the treatment, there's still a memory of that process, and the cells are still young by all means.
How do the surrounding cells interact with the de-aged muscle cells? Do you see any different cell-to-cell communications or interactions that weren't there before?
So those are we are doing those studies as we speak. So we anticipate that actually these cell autonomous rejuvenation will also have a cell non-autonomous effect. So this will have also obviously repercussions on the surrounding tissues. But we are trying to understand that process better. But I don't have a definitive answer yet.
And once you alter the cells, again, you don't know the interactions they're having with other cells. The rest of the mouse just seems to be as it was.
Well, again, so far we haven't looked at that question yet. The only thing that we know is that from a physiological standpoint, from a functional standpoint, the muscle where we transplanted the cells is much better. We haven't measured yet if there is also a systemic benefit by simply transplanting rejuvenated stem cells. So we don't know that yet. But it wouldn't be any different, in my opinion. But again, we need to prove that, or to study that in a little bit more depth. But it wouldn't be any different from the current technologies where you take, for example, young stem cells and you transplant young stem cells from a donor into a recipient. So again, in that case, you have, yes, truly young cells coming from a donor, and you're putting them into an older individual. So you obviously you would expect those cells to kind of communicate and interact with the host. I think the same principles apply here. So the only difference, which is a big difference, is that the cells are not coming from a donor, are coming from yourself. And so there is
right. I just wonder if you de-age. If you make one part of a creature's body youthful,
yeah,
it still needs to interact properly with all the other parts. In order to bring youth back to someone, you might have to de-age multiple parts of them, because if you only do one part, perhaps that part being more youthful will then put strain on the other parts, to the point where you get this this unfortunate trade-off, maybe that the organism would die just as fast or sooner because of a different complication. If I had a extremely young heart, but all the rest of me was old, perhaps that just wouldn't work. Maybe I need multiple parts to be youthful in order to function properly. And this, two parts being way out of sync wouldn't work. I don't know. Just a thought.
Oh yeah, sure. That is definitely a possibility, and it is possible that of course, we may have to intervene on different tissues. On the other hand, though, in some instances, it's the as we age, we increase the risk factor for diseases, for example, heart failure. Now, probably just by keeping the heart younger, that per se could actually prevent that from happening. And quite honestly, again, this is very early days, but it is possible that we may not have to. There could be some key tissues that could actually be the target, and then these one or two tissues on their own could actually be enough or sufficient actually for triggering a systemic rejuvenation. So, for example, it's known that the endothelium, or it's known that the hypothalamus, that there is kind of an endocrine and there is a regulation actually that is very systemic, and which is regulated by these key organs. And so if we could, for example, just rejuvenate the endothelium, so the entire vasculature, for example, that on its own could have a very strong effect systemically. And the same is true for other tissues. So again, this is very early days. So it is possible that we may have to work synergistically on many different organs and tissues. It's also possible that just by rejuvenating one major tissue, we may have a very strong effect systemically. So we don't know that yet.
Well, very good. What do you expect that you're going to be able to figure out over the next couple of years? What do you feel like is near at hand versus things that may take a long time to figure out?
I think that I'm very optimistic on the fact that in the relatively short future, some of the indications that we have identified, we will probably be ready for a clinical phase, for a clinical trial. And in particular, I'm thinking about sarcopenia, or muscle dysfunction, osteoarthritis, and some aging-associated conditions which affect the skin, for example, hair loss, hair discoloration, and wound healing. So I'm very positive that actually for one or more of these indications, we will soon have robust preclinical data actually to move forward. So this is in the short term. In the long term, I think that this technology can go beyond aging, and can actually be in a way applied to regenerative medicine in general. So again, if you extend the concept of aging to the fact that cells lose with time the ability of doing something, a specific function, for example, regenerating neurites or axons for the neurons, for example. If you could bring the clock back of that specific cell type to the point where the cells is still capable of regenerating the axons, then you can start thinking about many other implications and indications which have more to deal with regenerative medicine, rather than aging per se. And so I think that this is really a technology that could be broadly applied to a variety of different conditions that could really change the way we think about aging, but more in general regenerative medicine. And so I'm very excited.
Well, that's great. Well, Vittorio Sebastiano from Turn.bio, thanks for coming. I appreciate it.
Thank you so much for having me, Richard.
You've been listening to the Future Tech edition of the Finding Genius podcast. This podcast is information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed. Review us on iTunes or wherever you listen, and subscribe today by going to futuretech.findinggeniuspodcast.com.